In this lab, you're going to be testing the effect of enzyme concentration on reaction rate. So let's take a look at the chemical reaction and the enzyme that we're going to be using today in our experiment. So we're going to take something called starch, and starch is a polysaccharide. So a polysaccharide is it's a macromolecule, it's a polymer, and so it's made up of a whole bunch of these smaller subunits bonded together. And so in the reaction today, we are going to take starch and we are going to break it down into its individual components. So we're going to break it down into glucose. And glucose is a monosaccharide. So we're breaking these bonds here between the glucose molecules and starch to make these individual glucose molecules. And this reaction is going to be catalyzed by an enzyme known as amylase. And whenever we look at a chemical reaction like this, the enzyme is always written above the arrow. So I'm just going to point out here that starch is what we call the substrate, or you might also see it called a reactant. Glucose is our product, it's what's being produced, and amylase is our enzyme. Now, in order to have kind of a visual representation of what's happening during this reaction, we're going to use something called iodine. And when iodine is mixed with starch, so iodine is an indicator, and when it's mixed with starch, it makes it turn blue. All right, so starch and iodine mixed together looks blue. Glucose and iodine is clear. So we're going to be able to see this reaction happening because we're going to mix our enzyme with the starch, which is mixed with iodine, and we're going to most likely see it change from a blue color to a clear color. Now, to actually measure this change, because we want to be quantitative and not qualitative. So remember, quantitative means that we want numbers. We don't want to just say, oh, look, it's turning from blue to clear. We want to know exactly how much blue is in our sample and how quickly the blue is changing to the clear color. So we are going to use a machine or a tool called a spectrophotometer. And if we break down this word, we can kind of break it down into three parts. So spectro, photo, and meter. So meter means to measure. Photo means light. And spectro has to do with the spectrum of light. So we are specifically going to be looking at the part of the light spectrum that is made up of color. And we are specifically going to look for the color blue. All right, so the way that this spectrophotometer works is that we're going to put our sample in something called a cuvette. And a cuvette is a kind of a rectangular container like this. And so we're going to fill up our cuvette with our blue starch and iodine solution. And we're going to be mixing in the amylase as well. And inside the spectrophotometer, there is a light that's shining light through this sample. And so as this light moves through the sample, some of it gets absorbed or kind of stuck in the sample, and some of it passes through. And it kind of depends on how dark the blue is and which wavelength of light we are shining through. on the opposite side, there is a sensor. And this sensor measures how much of the light is transmitted through that sample. So it's actually going to give you a transmittance value. All right, and the spectrophotometer is going to be hooked up to a little mini handheld computer. And so when you put the sample, this cuvette, in the spectrophotometer, on your LabQuest unit, you're going to see a graph, and it's going to show you the change in color over time. So you're going to have time and transmittance here. All right, so by using our indicator of iodine and mixing our starch with our enzyme amylase, we can measure how quickly the blue changes to clear, and we can test different, we're going to look at different concentrations of enzymes. So we're actually going to look at 0% amylase, which is just water, and then we're also going to look at 0.25%, 0.5%, 1%, and 2%. And so on our graph down here, we're actually going to end up with multiple lines showing us the reaction rate of these different concentrations of the enzyme. And then we're going to do some analysis on that and then create a graph based on our analysis. So as we measure the reaction rate of these different enzyme concentrations, we're going to get the raw data that's going to show us the transmittance change over time. And we're going to end up with a series of lines. And so each line will correspond to a different enzyme concentration. So one might be 0% amylase, one might be 0.25%, 0.5%, etc., etc. And I honestly can't remember whether these lines go up or down, but you'll see multiple lines show up on a graph. Now, this is our raw data, and we typically don't want to present our raw data. So we're going to use the LabQuest unit, which is that kind of handheld computer, and we're actually going to look at the slope of the line. And the slope of the line is how much it changes over time. So by doing some analysis, we're going to end up with, you're going to see the equation of a line, which is y is equal to mx plus b. So this is just the equation of a line. And we're going to be recording this m value here. So this m value is the slope of the line, and we're using this as a way to measure the reaction rates. So you're going to end up with a data table where you have amylase concentration, and then you're going to have reaction rate, and this is your m value. And so it's going to look like at 0% amylase, you're going to have a number that you're going to write here. At 0.25%, you're going to have a number that you write here. 0.5, 1, and 2. So you're going to end up with a series of numbers here, and these are all just those m values. And then when you actually create your own graph of data, you're not graphing this raw data here. So you're not graphing the raw data. Instead, you're going to be graphing this analyzed data here. All right, so your graph should look like this. It should have your independent variable on the bottom and your dependent variable here. And then you can use your to write your title, you can use your formula you can use your formula to write out the effect of the independent variable on the dependent variable. And you're going to end up with a single line. All right, so remember you are you're graphing your analyzed data. You are not graphing your raw data. Today's procedure is time sensitive because as soon as you add the amylase to the starch, the reaction is going to start happening. And we want to keep this a controlled reaction, or a controlled experiment, I should say, because we're testing different concentrations of amylase. So remember, we're testing 0%, 0.25, 0.5, 1, and 2. So as we're collecting these 5 rounds of data, we want to make sure that we are collecting that data consistently. So let's start out by talking about how to collect data for 0%. So at the very beginning, you want to make sure that you have everything set up and ready to go. So you should have your test tube with amylase, your beaker that contains the starch and iodine solution, an empty cuvette, your spectrophotometer set up and ready to go, and it should be attached to your LabQuest unit, which has also been programmed and set up and ready to go. So once you have everything set up, one person is going to set a timer for 30 seconds. A second person at the same time is going to use a pipette, or you might be using something called a micropipette. It depends on what is available the day of the lab, but you'll be using one of these two tools, and you're going to use that pipette to pick up 500 microliters, which is equal to half a milliliter of the amylase solution. So you use the pipette to pull that into the pipette, and then use the pipette to transfer that over into the beaker containing the starch and iodine solution. Now, as soon as those solutions are mixed, then the timer needs to be started. So these two things need to happen at the same time, starting the timer and adding the solution into the beaker. Now, with the same pipette, you're going to mix for we'll say about 5 seconds. All right, so you don't have to mix it for very long, but about 5 seconds, just kind of swirl it around and mix it. And then you're going to use that pipette to, that's our second step. Our third step is to transfer some of that liquid into the cuvette. And we're going to fill it about 3/4 full. All right, so we're going to transfer that there. Our next step is to put our cuvette into our spectrophotometer. Our fifth step has to do when our timer reaches 0. When it hits that point, our fifth step is to press that play button. All right, so you have to wait until the timer reaches 0 before you select that start button. So you, after you start it, it's going to run the reaction and collect the data for the amount of time that you programmed it to. So you don't have to push anything to make it stop. It should stop automatically based on how you programmed it. So to reiterate, the key thing I want to point out here is that this 30 seconds is the amount of time you have to do all of this. All right, so for example, a lot of people want to mix it for 30 seconds, but you don't need to mix it for 30 seconds. You only need to do maybe 5 to 10 seconds because the cuvette needs to be in the spectrometer and you need to be ready to hit that play button when the timer hits 0. All right, so work quickly so that you can start your data at the right point. After you do this for the 0% amylase, you're going to do it for the 0.25% amylase. So same procedure, but you're going to use a different test tube. You're going to use the test tube labeled 0.25%. You're going to use the beaker labeled 0.25%, the cuvette labeled 0.25%, etc., etc.